Hi, I'm Kathy Hollis, and I'm the Collections Manager in the Department of Paleobiology at the Smithsonian Natural History Museum. Collections Manager is kind of like a librarian, but instead of caring for books, I help care for the vast fossil collections here in our department. And today, I'd like to talk with you about our digitization project and transcription for a project we're calling EPIC. That stands for the Eastern Pacific Invertebrates of the Cenozoic Collaborative. We're just one of about a dozen institutions that are part of this project, and how we need your help. Now I'm going to show you some specimens from our collection and what they look like, and then what the labels look like, and what we're asking you to transcribe. So we're now looking at one specimen from that drawer that we just saw. Every specimen in that drawer was collected from the same locality. And this was the tag that was created when it was collected. And it has information on it. It was United States Geological Survey of this particular district in Alaska, description of where it came out of the ground, the date it was collected. But the most important thing on this label is this number right here, 6695. And each specimen actually has that 6695, it's upside down, but it's written right there. So we know that 6695 refers to a point in the ground that's described in this label, but it's also described in various locality registers that we have in the museum. So when this was collected in 1913, the USGS scientist came back, and every place that was a locality was numbered, so 6693, 94, 95. And then this information right here is the most detailed information that we have that describes where every specimen in that drawer that we just saw was collected. So we want enough information transcribed by you from this label, especially this number here, to be able to tie back to the registers that we have in our collection, such as this one. Okay, here's another example of a label. So this is the locality number that we're asking you to transcribe, and I'm going to talk through a little bit about other important information on this label. So we know that this number, 18611, ties back to a locality register that has a description of where this object was collected. Here's a little bit more of that information, the locality information of where it was collected. There's also a collector name here, and in this memoranda field, it says California, but this field can be used for any sort of information, whatever the person filling out this label thought was important to note there. And then this here, Pecten elung, let's see, Pecten eldridger. So this is the genus and species name of this specimen. So this is very important information that we want you to transcribe as genus and as species. So this is an example of a label that is pretty complete. Here's an example of another kind of label. So we know that this is a USNM specimen, that stands for United States National Museum, locality number 14117. So this is the source, USNM, and 1147 is the locality number. And the label's a little different here, but we know that it's from the Kettleman Hills, California, so these are pre-made labels. And then we should be able to see, okay, because this is a very old label, this is the genus and species name here, and hopefully on the image that you're looking at, you'll be able to transcribe that. But we also want you to know everything else that's on this label, because the labels are so inconsistent, they're not standard, that really important information about what is in the collection could be anywhere written on the label. And I'll show you probably a really zany example. This is another picture of a label that we would be asking you to transcribe. And this is a field bag that the researcher, the collector, just wrote the notes down and just cut it and stuck it in the tray. But we know that this is a good label because it has the locality number 12462A. And as long as we have that number, like I said, we can tie it back to a register that has really complete data. And then the rest of this, because it's so unstructured, we would be asking you to just transcribe it verbatim. And then our collection staff here and our project staff here will be able to parse through what you transcribe and pull out the information and put it into a more structured format. Thanks again for your help on this project. I really look forward to collaborating with all of you.